Tardigrades, often called water bears or moss piglets, are microscopic invertebrates renowned for their ability to survive extreme conditions that would be lethal to most other life forms. Their resilience stems from a combination of physiological adaptations, biochemical mechanisms, and a unique survival state called cryptobiosis. Below is a detailed explanation of how tardigrades endure such harsh environments, including outer space, high radiation, and extreme temperatures ranging from 328 to 300 Celsius. Cryptobiosis, the key to extreme survival. Tardigrades' ability to survive extreme conditions is largely due to their capacity to enter a state called cryptobiosis, a reversible, a metabolic condition where metabolic activity is reduced to undetectable levels, often less than 0.01% of normal. In this state, tardigrades essentially shut down their biological processes to endure hostile environments. There are several forms of cryptobiosis, with anhydrobiosis, desiccation-induced, and cryobosis, is cold-induced, being the most relevant for their extreme survival. Anhydrobiosis, desiccation survival. When faced with dehydration, tardigrades lose up to 99% of their body water and enter a tun state, where they retract their legs, curl into a barrel-shaped form, and drastically reduce their metabolic activity. This tun state protects their cellular structures from damage due to water loss. Key mechanisms include trehalose production. Tardigrades synthesize trehalose, a sugar that replaces water in cells, stabilizing membranes and proteins by forming a glass-like matrix, vitrification. This prevents cellular structures from collapsing during desiccation. Disordered proteins, TDPs. Tardigrade-specific intrinsically disordered proteins, IDPs, protect cellular components by forming protective coatings around proteins and membranes, further stabilizing them during dehydration. Antioxidant enzymes. Tardigrades produce enzymes like superoxide dismutase and catalase to neutralize reactive oxygen species, ROS, which can accumulate during dehydration and rehydration, preventing oxidative damage. Cryobiosis, cold survival. In extremely low temperatures, tardigrades enter a similar metabolic state, halting biological processes to avoid damage from ice crystal formation. Their ability to survive freezing is enhanced by mechanisms like supercooling, lowering body fluid freezing points, and the production of cryoprotectants, e.g. glycerol, which prevent ice formation inside cells. 2. Surviving extreme temperatures. 328 degrees to 300 degrees. Tardigrades can tolerate temperatures from 328 degrees, 200 degrees, to 300 degrees, 109 degrees, far beyond what most organisms can endure. Their survival mechanisms include low temperatures, 328 degrees, cryobiosis and ice prevention. At temperatures as low as 328 degrees, close to absolute zero, tardigrades enter cryobiosis. Their cryoprotectants, like glycerol, lower the freezing point of their body fluids, preventing ice crystal formation that could rupture cells. Additionally, vitrification via trehalose or other molecules helps stabilize cellular structures during freezing. Experimental evidence. Studies have shown tardigrades surviving exposure to 196 degrees, 321 degrees in liquid nitrogen, and even 272 degrees, 458 degrees in liquid helium for extended periods. For example, experiments in 2007 demonstrated that tardigrades in the tun state could be revived after cooling to near absolute zero. High temperatures, 300 degrees, protein and membrane. Stabilization. At high temperatures, tardigrades rely on heat shock proteins, HSPs, and trehalose to stabilize proteins and prevent denaturation. These molecules act as molecular chaperones, maintaining protein structure under thermal stress. Limits of heat tolerance. While tardigrades can survive brief exposure to 300 to 100 degrees to 49 degrees, prolonged exposure, e.g. 24 hours at 151 degrees, is typically lethal. Their heat tolerance is enhanced in the tune state, where reduced water content minimizes thermal damage to cellular components. 3. Surviving outer space. Tardigrades are the only known animals to have survived direct exposure to the vacuum of space, as demonstrated in experiments like the TARDIS, Tardigrades in Space, mission conducted by the European Space Agency in 2007. Their ability to endure outer space involves surviving multiple stresses: vacuum, extreme cold, high radiation, and desiccation. Vacuum of space. The vacuum of space causes rapid dehydration, but tardigrades enter anhydrobiosis, forming the tone state to protect against cellular damage. Their ability to lose nearly all body water without dying is critical here. The tone state also protects against the absence of atmospheric pressure, which would otherwise cause physical damage to most organisms. Extreme cold in space. Space temperatures can drop to near absolute zero. Tardigrades' cryobiotic mechanisms, including cryoprotectants and vitrification, allow them to survive these conditions, as discussed above. Cosmic radiation. Space exposes organisms to intense cosmic and solar radiation, including ultraviolet, UV, and ionizing radiation. Tardigrades' radiation resistance, discussed below, enables them to withstand doses far higher than most organisms. In the TARDIS experiment, tardigrades survived exposure to solar UV radiation up to 1,000 times higher than Earth's surface levels. Experimental outcomes. In the TARDIS mission, tardigrades, e.g., Rictursius coronifer and Milnesium tardigradum, were exposed to space conditions for 10 days. Many survived and, upon rehydration on Earth, resumed normal activities, with some even reproducing successfully. 4. Radiation resistance. Tardigrades can withstand radiation doses up to 5,600 gray GI of gamma rays, compared to a lethal dose of 5 GI for humans. Their radiation resistance is attributed to several mechanisms. DNA repair mechanisms. Tardigrades possess highly efficient DNA repair systems. After radiation exposure, enzymes like dual glycosylases and polymerases repair single and double-strand DNA breaks. For example, the tardigrade Ramosauteus varieornatus has genes encoding DNA repair proteins that are upregulated during stress. D-soup protein, unique to some species. In R. varieornatus, a protein called damage suppressor, D-soup, binds to DNA, forming a protective shield that reduces radiation-induced damage. Experiments have shown that transferring the D-soup gene to human cells increases their radiation tolerance, highlighting its effectiveness. Antioxidant defenses. Radiation generates reactive oxygen species, ROS, which damage cells. Tardigrades' antioxidant enzymes, e.g., superoxide dismutase, catalase, neutralize ROS, protecting cellular components. Tune state protection. In the tone state, the compact structure and reduced metabolic activity minimize radiation damage by limiting the production of secondary ROS during exposure. 5. Other extreme conditions. Tardigrades can also survive other extreme conditions, showcasing their versatility. High pressure. They can endure pressures up to 6,000 atmospheres, 600 MPa, equivalent to depths greater than the Mariana Trench. Their compact tongue structure and robust cellular membranes prevent collapse under pressure. Toxic environments. Tardigrades can tolerate high concentrations of toxins, such as hydrogen sulfide or heavy metal, likely due to their ability to enter cryptobiosis and limit metabolic interactions with the environment. Starvation. In cryptobiosis, tardigrades can survive without food or water for decades, with some reviving after 30 years in a desiccated state. 6. Molecular and genetic adaptations. Recent genomic studies have revealed additional factors contributing to tardigrade resilience. Horizontal gene transfer. Some tardigrade species, like Hypsibius exemplaris, have acquired genes from bacteria and other organisms, enhancing stress tolerance. For example, genes for antioxidant production may have been acquired this way. Unique protein families. Tardigrades produce specific proteins, such as KHS, cytoplasmic abundant heat soluble, and SAHS, secretory abundant heat soluble proteins, which stabilize cellular structures during stress. Telomere maintenance. Tardigrades maintain their telomeres, DNA caps on chromosomes, potentially aiding long-term survival and repair after stress. 7. Limitations and vulnerabilities. While tardigrades are incredibly resilient, they are not invincible. Prolonged heat exposure. As noted, extended exposure to temperatures above 50 degrees is lethal. Active versus tuned state. Tardigrades are far more vulnerable in their active, hydrated state, where they cannot employ cryptobiotic protections. Rehydration risks. The process of exiting the tuned state requires careful rehydration. Improper conditions can lead to oxidative stress or cellular damage. Conclusion. Tardigrades' ability to survive extreme conditions, outer space, high radiation, and temperatures from 328 degrees to 300 degrees relies on their unique ability to enter cryptobiosis, coupled with molecular adaptations like trihalose production, disordered proteins, antioxidant defenses, and advanced DNA repair mechanisms. Their tuned state protects against desiccation, vacuum, and pressure, while cryoprotectants and heat shock proteins enable survival in extreme cold and heat. The discovery of unique proteins like ZUP and insights from genomic studies continue to reveal the molecular basis of their resilience, making tardigrades a model for studying extreme survival and potential applications in biotechnology, such as radiation-resistant materials or cryopreservation techniques.